Good morning, students. Today's topic is overlapping and split genes. So, coming for the introduction part, in prokaryotes, the coding sequences of genes are continuous. So, they will be having the sequences which is present will be only regularly going on with the productions. So, they will be having the genes which will be organized into a groups, and each will groups will have their own processing as transcription units, operons, and different genes will have different unit, and they will be separated from each other, and there is any overlapping process we have to see eukaryotic genes present. We can see some organizations such as split genes, overlapping genes, and pseudogenes. So we have genes which is nothing but long length sequences. So that will be split up into various names which will be helpful for their production activities. So coming for the codons, we already know about the genetic code. So it is a triplet code which will give you the sequences in a triplet codes, and which will give you the translation process and help you for the amino acid synthesis. So that is very important here, which is playing a role. So this is nature of the amino acid residues which will determine your proteins, and which is of your structures of your proteins also. So this was a study which has been carried out in the process. So although we generally say that the individual codons are mean for specific amino, so individual specific triplet codons will have their own specification of your amino acids and recognize the fact. So we have to be very clear with the codons. And this tRNA molecules, mRNA molecules, they are also very, very important for these processing. So it should be very clear. And the multiple points in this will have to be clear with the shape of the tRNAs and recognition of the tRNAs. All these things is very important in coding of this process. So which is helping for your folding of your protein. So these codons only will help you for the production of the proteins, which in turn will help you for the final production of your folding of your proteins. So it will give you the structure of the primary structure of your proteins. So this has been studied, and we have got many processing over this. So coming for the processing, in that the newly repeated code plays a key role in their later step in protein synthesis. So we have to talk about specification in certain things which will help you for the determining the structures. So in that case, we have to know about certain genes which will be having some process which is overlapping, or pseudo genes, or split genes, or multiple genes. So we have to know about what is these things. So coming for the overlapping gene, which is also a gene whose expressible nucleotide sequence partially overlap with the accessible nucleotide sequence of other. So one will be overlapping the other. So one sequence will be overlapping the other sequences, and which is finally will be determined only when you come to know about this processing. They may result problem in their sequences, codons, and finally the structure is also will be dependent. So there will be problem in their structure also. So this is very important. We have to talk about these things. So the function of that will be one or more gene products. So this is what we are talking in terms of overlapping genes. So coming for these bacteriophage, if you talk about that, it is containing a single stranded DNA, approximately 5,400 nucleotides in length, and genome which is of 9 criston. So we have plenty of things will be present over there, and we have the information which will code for the proteins, and they have to be very clear. So coming for these nucleotides where it is not exceeding the 6,000, so we should be nearly only 5,400 should be maintaining it. So the genes will be only that genes will be present. On a detailed study of the system will give you the basic arrangements of the segments and their cistrons and their proteins. So these overlapping cistrons will be theoretically possible if two cistrons have to function at different times, and the nucleotide sequence are translated in two different reading frames. So in case of this, they have undergone various things, and they got about this overlap. Cistron E is overlapping cistron D. So you have to say that a specific gene which is having the functions which is been lost due to some of the other sequences, which is nothing but it is overlapping the other things. So cistron E will be overlapping the cistron D. So this is a figure which will have you the various patterns of the gene arrangement, which will help you for the understanding. So this is a pattern where the cistron will be named. So A, B, C, D, E, F, G. It should be shown that number mutations in cistron E lie within the cistron D, and these mutation do not influence the translation of cistron D into proteins. So, do not influence the translation of cistron D into protein. So, cistron D is being affected. So, cistron E comes and do the work. So, cistron D cannot do their work because of the problems. They have overlapped enough the things. Similarly, some of the other nonsense mutations for cystone E is also tie up with the cystone D, suggesting that cystones D and E overlap in the DNA sequences, and that the cystone D is E are translated to different reading frames, so that the codon in mRNA of one cystone will not be read as a termination codon during the translation of mRNA of the other cystone. So there will be confusion in both these cystone D and cystone E, and finally they will be coming to know about there is some overlapping process, and the genes will conclude the processing and they wait for the other process to be held. So this will be known about the overlapping genes. The next we will be talking about the split genes. We have to know what is split genes. The genes with the interrupted sequences. So we have some sequences which is not in a continuous manner, and they have some problem that is split. They have the end, small end will be interrupted. Some sequence will be interrupted. That is will be known as your split genes. Usually genes means you should have a continuous sequences of nucleotides. In other words, we can also say there is no interruption in the nucleotide sequence of a gene. Such nucleotide sequences codes for a particular single polypeptide chain. And it is observed that the sequence of nucleotide was not contained. In case of some genes, the sequence of nucleotides were interrupted by intervening sequences. So few sequences will be intervening, that is disturbing of the other things which will be done. So split genes were independently discovered. The special thing that is split genes, which was discovered by Richard J. Roberts and Philip A. Sharp in 1977, for which they have shared the 1993 Nobel Prize in surgery or medicines. So the person who are talking about the interrupted genes, that is nothing but the split genes, and they were talking about the introns, exons, that is coding sequence and non-coding sequences. So we have to know about these sequences. Normal sequences will be exons, and interrupted sequences is nothing but introns. Normal sequences is exons, and interrupted sequence will be introns. So we have to be very clear. So we have to have a normal sequence which is having the production level for the next things. So transcription, translation, finally we have to go for the production of your protein. So the sequences should be in a proper way. Only coding sequences should be present. No non-coding sequences should be present. So this is representing the nucleotides which will be translated to form a DNA, and they will be very helpful for this thing. So coming for these split genes, they are the intervening sequences, that is this yellow color implies that is introns. They are not needed for coding for the next processing. So they are just intervening sequences. So they are going for the coding, they will give you some other changes in their gene processing, and finally the proteins will not be formed. So coming for this picture, so we have this yellow color which will be intron, so that will has to be removed. Only the coding regions will be undergone. Transcription, splicing. So this is your normal picture which will help you for the introns, and then splicing process, and removing the introns. So interrupted sequence has to be removed. Only then you have to get a proper things which will be coding for the production of the protein structure. So we will have the mRNA which will be functioning mRNA by addition of some poly A tails and 5' groups. So remove all your introns, only that exons will be present. So this exon is yellow color, this intron is light red color. So this has to be removed, and only the full yellow color has to be done, and that will be used for the production. So coming for this process, the intervening or interrupted sequence of split genes are known as introns. So these sequences do not code for any peptide chain. So they are not needed for the coding process. So they have the special role. So they should not, they will be not useful for the transcription. So interrupted sequence are not included into mRNA, which will be transcribed from DNA of the split genes. So important features of split genes. So what will be the role of the split genes? They have to be important role. As each interrupted gene begins with the exons and ends with an exons, and the exons occur in the same precise order in the mRNA in which they occur in the gene. The same interrupted gene organization is constantly present in all the tissues of organization, and introns are blocked in all the reading frames. So these are very important things. They have to be undergone only exons, not needed introns. So termination codons and everything will be, most introns are not seen for having coding functions. So this is having some special role, like they help for the eukaryotic genes, and also some of them are active regions for the protein molecules in case of some of the antibodies productions. And introns and also evolutionary process, they will be helpful for producing new larger genes. It is also possible that some introns have been introduced with certain exons during evolutions. And when you talk about the introns, may also provide for increased recombination rate. So this introns, not only intervening sequences, are helpful for the recombination rate between exons of a gene, and they may be of some significance in genetic variation. Introns are known to be code for enzymes involved in the processing of HRNA. So these are the significance of the split genes. So today we are talking about this overlapping genes and split genes. So finally it was over. Thank you.